I'm John Fulmer, a student with the University of Washington's Engage program, here to welcome you to our event tonight. Stella Stylianidou is a physics PhD student at the University of Washington, pursuing research in biophysics. She studies traffic paths of molecules inside bacteria and how bacteria manage to send those molecules to specific locations before they split into two cells. Please give a warm welcome to Stella.
Thank you for that lovely introduction. I would like to take you on a trip today inside a bacterium. And I would like together to figure out how does the bacterium measure distance and how does it send molecules inside its body to specific places in the cell. You may all know that we're made out of cells. We're made out of these building blocks. There's something like Legos that build our body. And we have a lot of types of cells. We have different types of cells, such as heart cells or brain cells. And they're all different and they do different functions. But all of them together work for our body. And each one of them is independent and separates independently. We have a lot of those cells. We have 10 trillion human cells in our body. But you may not know that we have even more bacteria. We have 10 times more bacteria in our body. We have 100 trillion bacteria in our body. You're now carrying 5 pounds of bacteria on you. And so you may think of yourselves as humans, but I think of you as mostly bacteria. You may have heard of bacteria as germs, bugs, or microbes. And there are different species of bacteria, like there are different species of animals. And they come in different shapes, like spherical, rod-shaped, or spiral. They're really, really small. They're so small that you can fit 1,000 of these guys on a pin. So you may be wondering, why do we study bacteria? Why should we care? The way I see it, there are 3 reasons to study anything. They're good for us, they're bad for us, or they're just interesting. And I think bacteria fit all these 3 reasons. First of all, they're bad. You may have heard of E. coli, Salmonella, Streptococcus, or cholera. And if you know about them, it's because they're bad. They gave you some infection, maybe they gave you food poisoning. And then you went to the doctor and he gave you antibiotics to kill these bad bacteria and become healthy again. And you may know that a lot of bacteria are becoming resistant to all the antibiotics we have. So we need new ways to fight them. We need to understand them better and figure out how we can make new antibiotics to fight these bad guys. And you may be wondering, okay, why do I have 100 trillion of these bad guys in my body? Can we take them out? I don't really want them. Well, they're also good. Your body has outsourced a lot of the things it needs to do to these good guys. They produce vitamins for you, they produce minerals for you, and they also train your body to fight the bad guys. They teach your immune system how to distinguish friends from foes. Furthermore, we've been recently learning that they're related to even more things about our health, like obesity, chronic disorders, and allergies. We've been figuring out that the diversity of bacteria we have in our body can be related to these things. For example, when you take stool samples from a fat mice and put it on a thin mice, the thin mice becomes fat. So the type of bacteria you have in your body can be related to whether you're obese or lean. And we're still learning a lot about this. It's a new research. Last, they're interesting. They're simple. They're simpler than our cells, so we can use them to learn about our cells. But they're also very complex. They're so interesting that they have such interesting ways to make sure their offspring survive. And that's what we're going to talk about today. So this is our character. This is our little friend. His name is E. coli. He was born a few minutes ago. He lives in your gut, but it's okay. He's one of the good guys. And he likes to eat the sugars you eat. And he was nice enough to let us look inside, so that's how the inside looks. Around it, there is a wall that protects the inside from the outside. It has those little hairs sticking out that help it stick to surfaces, like your gut. At the end, it has those tails, and that's how it goes towards food. So it can smell food, and then it uses the tails to move there. And then in the middle, the yellow spaghetti you see is the DNA. And the DNA in the bacterium is really compact, so condensed that if I took it and unfolded it, it would be 1,000 times longer than the bacterium itself. But what is the DNA? Well, it's an instruction manual. It tells the bacterium how to make bacteria and how to make every single thing inside its body. How to make all these little molecules that you see here that are called proteins. And these proteins are machines. They're machines that the cell uses to build everything, like its cell wall, and also to produce energy or eat its food. So it has all these machines inside its body that makes from the DNA. So while we've been talking, our little friend has been growing. And you probably know how humans make babies. Well, bacteria do it in a slightly more boring way. They just grow, and then they split in the middle. And as we said, the bacterium has this piece of DNA inside its body. And of course, it can't leave one of the babies without DNA, without the instruction manual on how to make everything inside your body. So it has to copy that piece of DNA and make 2 identical instruction manuals that have every single word. And then it gives one to each cell. Now, imagine for a bit that our bacterium did not split in the middle. What would happen? Well, one of the babies may not take the piece of DNA and it may die. So it's really important for the bacterium to split in the middle. And it manages to do that very accurately every single time. It somehow finds where the middle is and it splits there. How does it do that? Does it have a measuring tape? Does it measure where the middle is? What kind of ruler does it use to figure out where the middle is? Well, we said that it has these machines, these proteins inside its body that do everything it needs. So it probably uses one of these machines. Since it doesn't have a measuring tape, it probably uses a protein. So it would be really useful if we could look inside the cell and see which protein sticks in the middle and helps the cell split there.
What's that word?
Ruler?
No. Protein.
Protein. Yeah.
Oh, I didn't hear that. Thank you.
So it would be great if we could see inside. But as you can see here, we can see the bacteria pretty well, but we can't really see inside. We can't see any of the proteins that are inside the bacterium cell. So we were kind of stuck about figuring out where the middle is and how the bacterium cell finds the middle. But there was this professor in Friday Harbor that studied jellyfish, and he found this jellyfish that had a green glow around its rim. And he really wanted to know why that jellyfish glowed green. How did it do it? Why did it do it? So he took thousands of jellyfish and squeezed them and took out what he called the squeezate. And he took it to the lab to study it and figure out why it was green. What he found was that the jellyfish made this protein. This protein was fluorescent, which means it absorbs light of one color and it emits light of a different color. In this case, it absorbed blue and emitted green. He didn't understand at the time how important that was and how important that would be for biology. But 30 years later, we realized that if we take the instructions on how to make the fluorescent protein from the jellyfish and put them in the bacterial DNA, then our bacteria would also be green. And even better, if we put that instruction of the fluorescent protein next to a protein we care about, then we could see where the protein is. It would be like dressing our proteins with glow-in-the-dark t-shirts, and then we can see them. And the best thing about this is all you have to do is put these instructions in the DNA, and then the cell does it for you. It makes these proteins with glow-in-the-dark t-shirts. So now all you have to do is shine some blue lights to the cells. The fluorescent proteins absorb this blue light, and you see green spots wherever these proteins are. Let's see how that looks inside a cell. As you can see, things are not very stationary and peaceful. They're moving up and down, they're bouncing off of other things. So it's surprising that the bacterium manages to do anything with how not peaceful things are in there. So now we attached these fluorescent proteins to several different proteins, and we found one that's in the middle. And it seemed to make a ring. It seemed to make a ring around the middle, close to the membrane, and then close to the splitting time, the ring would become smaller and help the cells to split. So this is how it actually looks. You can see the green lines in the middle, and that's where the cell splits. And as soon as the cell splits, the green just goes everywhere in the cell, it spreads around, and then again it goes in the middle and makes this new cell to split. So what happens there is that I have these ring proteins that go and attach in the middle, and they attach to each other and they make this ring structure. Then this ring closes and draws the membrane together and helps the cell to split. But that still didn't tell us how this thing knew to go in the middle. How did this ring knew where the middle was and went there and just made this ring there to make the cell split? Well, in biology there is this main tool. We take the DNA, we take this instruction manual, we delete a page, and we see what happens. You can think of it as taking the IKEA manual how to make chairs and deleting a phrase, and then if you see that the legs are at the wrong places, you knew that that phrase was really important on where to place the legs. So in this case, if we just deleted a few instructions from the DNA and we deleted some instructions for a protein, and we saw that the cell didn't split in the middle anymore, we would knew that that was the instruction, that was the protein that told the ring to go in the middle. So we started deleting the instructions, and we found that there was this protein that when we deleted it, it would make these mini cells. The cell wouldn't split in the middle anymore. And we named that protein min-C because when it's deleted, it makes mini cells. But how does it do it? How does this protein talk to the ring and tell it go in the middle? Well, we have a tool to figure it out. We can attach to this protein a glow-in-the-dark t-shirt and see what it does in the cell. And when we do that, we see that our protein is moving up and down from side to side. Or if you want to see it in a snapshot, it's from one side, then it goes to the bottom, and then again at the top, then again at the bottom, and again at the top. And it doesn't seem like it spends a lot of time in the middle. It spends most of its time at the two sides. You can think of it as a person running up and down the field. He runs really fast, he gets to the end, now he has to slow down, change directions. And he runs really fast, he gets to the other end of the field, he has to slow down and change directions again. So this guy will be spending the least amount of time in the middle because he doesn't have to change directions there. And that's what the min protein is doing. It's moving really fast, it has to go to the end, and then it has to change directions. Then it moves again fast, and it has to go to the end and change directions. So it spends the least amount of time in the middle. But what does that mean? Well, it ends up being that the ring and this min-C protein are not very good friends. They kind of hate each other. When you put them together, they don't interact very well. When you have this min-C protein, the ring is just spread around. It can't attach the proteins together and make this ring structure. But in the places where you don't have min-C protein, then you can have the proteins attaching together and make this ring structure. And because, as we said, this min-C protein moves from side to side and it spends the least amount of time in the middle, the ring can only form this ring in the middle, because that's the place where this min-C protein is the least found. So this is our ruler. That's how the bacterium knows where the middle is, and that's how it puts the ring there. It uses this mean C protein that goes from side to side, and it spends the least amount of the middle. So that's how our little friend finds the middle. And finding the middle is one of the most important things bacteria have to do. As we said, if they didn't split in the middle, one of the babies would die. But there are other places they have to find. They also have to know where the edges are. And they also have to know which edge is where. For example, sometimes they send proteins to only one side of the cell. And I've been talking like bacteria are these youthful new babies that are always young. It's not exactly true. And the way they do that is that they send the junk proteins to one side. So they sacrifice one of the babies to keep the other one young. So it needs to know where the one side is to be able to push things there. How does it do that? How does it measure distance and know where one side is? It also sometimes sends things to both sides. Again, how does it know where the two sides are? How does it do that? And then sometimes it sends things to the middle. So that's what I studied. That's what I care about. And if I wanted to figure out how does it send things to the edges, I could just look at a protein that goes to the edges and look at its motion and see if I can figure out why it goes there. The problem is that the cell breaks down proteins after they're used, and it may have more active ways to put them there. It may have some machines to put them there. So instead, I decided to use something that is not usually found in the cell, something that's kind of like a useless beacon, to see what it would do and where would it go, so that it wouldn't be broken down by the cell and I would be able to watch it for a long time. So I put this thing in the DNA of the bacterium, and then I put next to it our fluorescent t-shirt, and then I looked at how it moved around. And then I looked at a lot of those useless beacons to see how they moved around on average in general. So when I looked at the first box, I saw that they were pushed towards that point on average. And then when I looked at the second box, I saw that they were pushed in the middle of that box again. And then when I looked next to that, I saw that they were again pushed in the middle of that box. So according to where you were in the cell, you seem to be pushed at 3 specific points, which was pretty weird. So how did that happen? Well, there's this idea out there that the DNA of the cell, by being really compact and by taking space inside the cell and not allowing things to move through it, can actually push things to the spaces that there is no DNA. You can think of it as a twin peak mountain. If you're my little protein moving around and you're at the two edges, you're not really a hiker. You don't really want to climb the mountain. You're just moving up and down, bouncing up and forth. You're probably going to end up at the valleys, because that's easier. And then if you were one of those hikers that made it above the mountain, and then again, you're probably going to end up at the valley, because you're not trying to hike the next mountain. You just ended up there. So we think that the DNA acts in such a way, by taking space in the cell, acts like two mountains, pushing things in all the other regions that do not have the DNA. I want to finish my talk by summarizing of what our little bacterium uses. So we said that the way it measures where the middle is by using this mean C protein that goes from side to side, and that's how it tells the ring to go to the middle. And it also uses the DNA to send things at the regions that the DNA is not, so at the ends of the cell. Finishing up, Jacques Monod said that once we understand the biology of E. coli, we'll understand the biology of an elephant. E. coli is one of the simplest organisms out there, and we don't understand it very well. So by understanding the simplest cells out there, we'll be able to understand our cells better, elephants better, humans better. So we're still learning a lot about how E. coli works. We're still learning a lot about bacteria. We're still learning a lot about how they measure distance in their cells. And hopefully one day we'll understand E. coli very well, so we'll understand elephants and humans. I would like to thank the Weekends Lab that taught me all the biology I know, the Engage program for helping me give a better talk and not so boring talk, and the College of the Environment and the Town Hall of Seattle for hosting us here. I would be happy to take any questions you may have.
Thank you very much for that wonderful talk and explanation. Can you speak, I just had a couple of questions. The MinC that was bouncing back and forth from one end of the cell to the other, what kind of time frame was that occurring, that cycle? And also, can you speak a little bit to the process by which the molecule, protein rather, that actually does the cleaving in the middle, how does that know and how does that go about actually shrinking to do that cleaving? Thank you.
So your question was about the time frame of MinC and about the cleaving in the middle. The time frame is in the frame of minutes, but I'm not exactly sure. And I'm not sure if we know exactly how the cleaving in the middle happens. We know that it attaches to the membrane and it somehow draws it in. It has to do with the fact that the DNA is there. So while you have the DNA there, you can't have the cleaving happen. So the ring has to wait for the DNA to separate for the cleaving to happen. So we know that it needs to wait for the DNA to separate. But I'm not sure we know exactly how it happens.
So you said the DNA, when you put in the new proteins of your own, that DNA kind of stops it from going to certain places. Why doesn't that happen with the MinC? Is there something special about the MinC that makes it go through DNA?
It has to do with the size of the protein. Like bigger proteins are more likely to end up to be pushed by the DNA at the edges, whereas the MinC doesn't do that. But there's more interactions going on with MinC that make it go from side to side, whereas what I put there was just a useless protein that had no other function.
How do you put something into DNA?
That's an excellent question. I wondered the same too when I joined the lab. I thought it was very surprising that we can do that. Viruses in bacteria have ways to put their DNA in the bacterium and cut the DNA in bacterium and attach their DNA in the middle. So we use viruses to do that and to put DNA in bacteria. We can also put small pieces of DNA that are not attached to the main big piece, and that works too. The bacterium will still read that piece of DNA and make the proteins that we want it to make.
I have a question, just real quick. It might not have been related to your study, but I heard that you mentioned that if you introduce bacteria from fat mice into skinny mice, and skinny mice will become fat,
Yeah.
would the same things become vice versa? You introduce the skinny mice bacteria into a fat mice, would the fat mice become skinny?
I think it does, yeah.
When you talked about the DNA acting like mountains, pushing the proteins to the sides, what prevents the DNA from moving around? Is there something holding it in place? Or, I mean, how do you know that the DNA is not going to move? And
It's a good question. There are some proteins that make the DNA be really condensed and be really crowded in the middle. So maybe that's what's keeping it there, but I'm not sure.
So the MinC is when you have the absence of the MinC, that's when you have the ring proteins form, right? The rings form in the absence of MinC. And then when you don't have MinC at all, you have rings form at the ends, right? Okay. And then okay. But I guess the other question I had was how many tries did it take before you identified MinC specifically?
So I didn't do the work about MinC. I was talking about the scientists. Somebody else did the work about MinC.
Okay.
So I don't know how many tries it took them to find it. Probably a lot. Probably we identified a lot of things and deleted a lot of things. But that's how we know what the DNA bacterium does. They deleted a lot of things and they would like, okay, what happens when I delete this section? And that's how they figured out what the DNA does. But yeah, wherever you don't have MinC, you get the ring forming. And when you take MinC out of the cells, then the ring can form anywhere. So you get mini cells.
So the follow-up question, I guess, would be, what part of it was your work?
The DNA part with the mountains.
Okay.
Yeah. More questions? Okay. Thanks a lot.